Welcome back to Studious students. In this video, we'll understand tetanus, mainly pathogenesis, transmission, clinical symptoms, lab diagnosis, treatment, and prevention of tetanus. So let's begin with the pathogenesis of tetanus. Clostridium tetani produces two important substances, tetanolysin and tetanus toxin. Tetanolysin has no role in pathogenesis, whereas tetanus toxin is responsible for disease symptoms. Now, let us understand the mechanism of tetanus toxin. Clostridium tetani produces tetanus toxin, which travels to the central nervous system. Normally, inhibitory neurotransmitters like GABA and Glycine control excessive muscle activity, but tetanus toxin blocks the release of these inhibitory neurotransmitters. As a result, excitatory neurons keep stimulating the motor neurons continuously. This causes continuous muscle contraction, muscle rigidity, and painful spasms that are characteristic of tetanus. Tetanus is not spread from person to person. It occurs when Clostridium tetani spores enter the body through contaminated wounds or infected sites. Common modes of transmissions include injury or puncture wounds contaminated with soil or dirt, surgical procedures performed without proper aseptic precautions, infection following delivery or unsafe abortion, especially neonatal tetanus, and sometimes chronic infections like otitis media can also act as a source. Now, coming to the clinical manifestations of tetanus, the first and the most characteristic symptom is lockjaw, also known as trismus. Due to continuous muscle contraction, the muscles of jaw become rigid and painful. Because of this, the patient finds it difficult to open the mouth properly. This is usually one of the earliest signs that raises suspicion of tetanus. Another important symptom is difficulty in swallowing, also called dysphagia. Due to spasms and rigidity of the throat and swallowing muscles, the patient experiences pain and difficulty while swallowing food or even liquids. This symptom usually appears as the muscle rigidity progresses and can interfere with proper nutrition and hydration. As the disease progresses, the patient develops severe muscle stiffness and painful muscle spasms. These spasms occur because the muscle remains continuously stimulated without proper inhibition. Even minor stimuli like light, sound, or touch can trigger sudden painful spasms. Another classical feature of tetanus is rhizus sardonicus. It refers to the abnormal fixed screen-like facial expression caused by spasm of the facial muscle. Because of the continuous muscle contraction, the corners of the mouth are pulled outwards, producing this characteristic expression. In severe tetanus, the patient may develop opisthottomus position. This occurs due to general spastic contraction of the extensor muscles of the back. As a result, the body becomes arched backwards in a characteristic bow-like position. Now coming to the lab diagnosis, for specimen collection, samples are taken from the necrotic depths of wounds. Superficial samples are usually avoided because the organism is more likely to be present in the deeper necrotic area. On gram staining, Clostridium tetani appears as a slender gram-positive bacilli with a characteristic round terminus spore. Because the terminus spore bulges at one end, the organism gives a typical drumstick appearance. For culture, the specimen is inoculated into Robinson Cook meat broth, which provides an anaerobic environment suitable for growth of Clostridium tetani. On incubation, the broth becomes black and turbid due to bacterial growth and protein breakdown. Clostridium tetani can also be cultured on blood agar containing polymyxin B under anaerobic conditions. On blood agar, the organism shows characteristic swarming growth. This produces concentrated swarming rings. Toxin assay helps in detecting tetanus toxin produced by Clostridium tetani. It can be performed by in vivo mouse inoculation test on specimens such as serum and urine. Prevention of tetanus can be done by passive immunization using tetanus immunoglobulins. There are two preparations: human tetanus immunoglobulin and anti-tetanus serum. Human tetanus immunoglobulin is given as 250 international units intramuscularly, whereas anti-tetanus serum is given 1500 international units IM. Here's the main difference between human tetanus immunoglobulin and serum anti-tetanus. Human tetanus immunoglobulin gives about 30 days of protection, whereas serum anti-tetanus gives about 7 to 10 days of protection. For complete protection against tetanus, both active and passive immunization are given together. Active immunization is provided using TT or TD vaccine in one arm, whereas passive immunization is provided using human tetanus immunoglobulins or anti-tetanus serum in the other arm. Passive immunization gives immediate protection by neutralizing circulating toxin, while active immunization helps the body develop long-term immunity. Giving them in separate arms prevents interference between the antibody preparation and the vaccine response. Antibiotics are given to eliminate Clostridium tetani from the wound and to stop further toxin production. The drug most commonly used is metronidazole for 7 to 10 days. Alternatives are penicillin and doxycycline. Active immunization against tetanus is done using tetanus toxoid. Common vaccines containing tetanus toxoid include TPD vaccine, TD vaccine, and pentavalent vaccine. Now let's look at the immunization schedule for children. Pentavalent vaccine is given at 6 weeks, 10 weeks, and at 14 weeks of age. Booster doses are given at 16 to 24 months and again at 5 years of age. Additional doses are given at 10 and 16 years of age. Thank you.